Welcome dear students. Welcome to another tutorial from Brilliant Bios. In this class I am going to continue my vesicular trafficking lecture. In my first class of vesicular trafficking lecture, I have discussed about how protein glycosylation modification occur during this process. If you have not watched my first video, please watch the first video, then watch the second part of this vesicular trafficking lecture. In my first video, I have mentioned that this process, this vesicular trafficking, is nothing but secretory pathway of proteins. Why it is called secretory pathway? Because in this process, the protein are destined to different location, and one of the destination is cell exterior. That means the protein will be secreted out. So this process is, the name is given secretory pathway. This is also known as vesicular trafficking because during this secretory pathway, the process actually involved by the vesicles. So in this class, I will give you the very basic idea about this process. The first two things you have to understand in this vesicular trafficking topic. The first is vesicle budding. So there are two mechanisms are involved during this vesicular trafficking. Only two mechanisms. If you have understood these two mechanisms, you have totally understood the vesicular trafficking lecture or topic. The first is vesicle budding and the second is vesicle fusion. These two things repeatedly occur in this total process. I am giving you an example. When a protein enters inside the ear, I have discussed about in my protein sorting class. This is another thing. But from the ear to the cis-Golgi network, from the cis-Golgi network to the cis-cisterna, from the cis-cisterna to medial to trans to trans-Golgi network, and ultimate to these destinations. This total, all these events occurs only by these two ways. One is budding, one is fusion. So when protein enter inside the ear, then from the ear, the protein will enter inside the cis-Golgi network. Then vesicle budding occur from the ear, and then fusion will occur with the cis-Golgi network. So vesicles look like this, round or circular. This vesicle is nothing but plasma membrane of the endoplasmic reticulum. So this is called vesicle. In this total vesicular trafficking process, two things occur. One is budding, the vesicle bud from the ear and then fuse with the cis-Golgi network. And the same thing will also occur. The vesicle will bud from the cis-Golgi network or CGN and fuse with the cis-cisterna. The vesicle will bud from the cis-cisterna, fuse with the medial. Vesicle will bud from the medial, then fuse. The same repeated thing occur. So if you have understood the vesicle budding and fusion, you will understand the total mechanism. Now in this class, I only give you the idea of vesicle budding, how this vesicle budding occur. In this vesicle budding, two major proteins are responsible. The first major protein is called GTP binding protein. This protein actually helps to trigger the budding. The second thing is, the second examples or second proteins that is very important are called coat protein. Coat protein. As I have mentioned that vesicles are nothing but plasma membranes, so they are very delicate and fragile. During their transport from ear to Golgi, they can be fragmented. So they should be protected during this movement. And the protection is given by coat protein. Yes, another type of example of protein is very important for the budding is called dynamin. I will also discuss about this protein function, dynamin. So there are three proteins responsible for only this vesicle budding. In my next lecture, I will talk about this vesicle fusion, how vesicle fusion occurs. This is very important topic also. Now, in case of GTP binding protein, there are two types of protein are there. Examples, one is called SAR1 and another is called ARF. These two examples are only there for GTP binding protein, and I will discuss how this GTP binding protein helps in the budding. The next is coat proteins. Examples are COP1, COP2 and clathrin. Okay, there are 3 examples of this coat protein, and the dynamine is a protein that actually perform GTP hydrolysis. GTP hydrolysis. So I will talk about this in later. So first thing is protein but from the ear to the cis-Golgi network. This is the first route, and in this route, when protein actually move from ear to the cis-Golgi, this SAR1 protein is actually helps in this budding. So in this case, only for ear to the cis-Golgi, SAR1 GTP binding protein is important, and the coat protein among them, the COP2 helps in this budding. You have to remember this. SAR1 and COP2 help in the budding of ear, from the ear to the cis-Golgi network, from the ear. Okay. Now the SAR1 protein is actually a GTP binding protein. When SAR1 protein is bind with GTP, it actually helps this coat protein to cover the vesicles. But the SAR1 proteins has a intrinsic GTPase activity by which it can degrade the GTP into GTP, and when the SAR1 is attached with GTP, it will actually dissociate all the coat protein. For the better visualizations, I will refer 2 books. One is Lodis and Bruce Albert. So during this process, during this budding, SAR1 and COP2 helps in this budding process. Okay, now this is called, this type of movement from the ear to Golgi called anterograde movement. Okay, this movement is called anterograde movement, or very easily you can say forward movement, because the protein is moving forward from ear to the Golgi. Okay, so you can say in short or easily forward movement, anterograde or forward movement. But another thing can occur, that is sometime some endoplasmic reticulum proteins are wrongly sorted inside the vesicles. Wrongly sorted. Some endoplasmic reticulum protein that should be present in the only endoplasmic reticulum can come out in these vesicles and can fuse with the cis-Golgi network. Then this protein that is actually present only in the endoplasmic reticulum should be retrieved from the cis-Golgi to the ear. If it is wrongly present in the cis cisterna, then the protein should come out from the cis cisterna to cis-Golgi, then cis-Golgi to ear. This type of movement called retrograde movement, backward movement. How it occurs, I will tell you. Let's suppose if some endoplasmic reticulum protein enters inside the cis cisterna, that should not be present in the CGN. But if some proteins enter, then the protein should be retrieved back again to the endoplasmic reticulum. The same is true if the protein of the endoplasmic reticulum somehow, let's assume that reach to the trans-Golgi or trans-Golgi network or this medial or cis cisterna. What happens? These are the ER proteins. The green mark are the ER proteins. Then retrograde movement occur. This protein will be retrieved from this trans-TGN to the trans to the trans-Golgi network to the trans, then trans to the medial, and from medial to the trans, cis, and then cis from the cis cisterna, and then ultimate to the ear. This retrieval mechanism also present in this vesicular trafficking. So this is called retrieval, or you can say it retrograde movement. Retrograde movement. And in this retrograde movement, what happens? Which type of GTP binding are responsible? ARF GTP binding protein is responsible. And what type of coat protein is responsible? That is COP1. You must remember this. Here COP2, COP1. Now you can ask me, what type of vesicles actually bud out by the help of COP2? The proteins that are distinct for the lysosome. Let's assume some lysosomal enzymes that should be sorted out inside the lysosome, they can go through this COP2 anterograde movement. But some wrongfully, some endoplasmic reticulum proteins also enter inside the vesicles. So they should be retrieved. So let's suppose these red color proteins are lysosomal proteins. If these lysosomal proteins enter this site, this is totally right. These vesicles will fuse with the endosome or directly to the lysosome. This is totally good. But if this green endoplasmic reticulum protein enter inside this Golgi, they should be retrieved because they should not be present in this Golgi complex. That retrieval process is occurred by the retrograde movement by the help of ARF and COP1 protein. Now, another thing is that is when the protein that are destined to the endosome, lysosome, plasma membrane or cell exterior reach to this last border, that is called TGN, trans-Golgi network, then the budding occur. Then in that budding, in this budding process, this ARF protein actually helps in the budding. Only in case of ear to the cis-Golgi network, the SAR1 is responsible, and COP2 protein is helping the coating, and COP1 for retrograde movement. And in case of this movement to the endosome or to the lysosome or plasma membrane to the endosome, I will talk about one coat protein is important, that is called clathrin. Clathrin protein, the shape of this protein is called triskelion. It is called triskelion because it has 3 heavy chain, 3 light chain. Okay, so here when proteins come out from the TGN to the endosome with the help of clathrin, but during the direct shorting from the TGN to the lysosome, clathrin protein actually not involved, because clathrin protein requires an adapter protein. We call adapter protein, we name it AP2, AP3, AP1. So in this way, the clathrin protein requires adapter protein. But when proteins enter directly to the lysosome, there is no requirement of clathrin. Only AP3 is responsible for the direct sorting of protein to the lysosome. But another thing occurred, that is at first the protein entered inside the endosome, and then the endosome actually fuse with the lysosome. In that way, clathrin and AP1 protein is important. Okay. Now another thing can occur, that is if some cells can perform endocytosis, and if this endocytosis occur from the plasma membrane, the protein can enter at first inside the endosome, then the endosome will fuse with the lysosome, and in this process, plasma membrane to the endosome, clathrin protein also responsible. So from the TGN to the endosome, clathrin important. From the plasma to endosome, clathrin important. And when the protein will come out from the cell to the exterior, also clathrin is important. So clathrin is also here present, clathrin and AP2. This is adapter protein, helper protein of this protein. Okay, another important thing I have forgot to mention, that is during this retrograde movement or retrieval process, there are so many signal sequences are present that can be actually identified for this retrieval movement. I will give you only 2 examples that are so important. One signal is called KDEL. K for leucine, D for aspartic acid, E for glutamate, and L for lysine. So K means, these are the amino acid sequence. K means leucine, D for aspartic acid, E for glutamate, and L for lysine. Endoplasmic reticulum proteins that are soluble in nature. So soluble ER protein Only soluble ER proteins have this sequence, KDEL. You have to understand this. There are so many compartments, Golgi, endosome, lysosomes. This protein, these resident proteins don't have this KDEL. These are the resident proteins. So resident, that is, this protein should be present only in the endoplasmic reticulum. But if it is wrongly shorted into the cis-Golgi network or this Golgi complex, then it should be retrieved. And there is a signal sequence that is called KDEL. Only these resident soluble endoplasmic reticulum protein have this sequence. The resident membrane bound ER protein don't have this KDEL. Membrane bound, those proteins are membranous. Let's assume this is the endoplasmic reticulum. If the protein is membrane bound, then this protein don't have KDEL. Those proteins that are present inside the endoplasmic reticulum in soluble conditions, they have KDEL. Membrane bound proteins have different sequence called KKXX, diacidic, diarginine sequence. I'm not going to this this way, but KDEL is present in the soluble endoplasmic reticulum, and this is very important because this KDEL is actually a retrieval signal, and this receptor for this KDEL is present in the cis-Golgi network. You must remember here the receptor is present. So at first this receptor can sense that this is not the protein of this CGN, not to destined for this compartment, but it is actually wrongly have come to my side. So it should be retrieved, or should be retrograde movement, so that the protein can reach inside the endoplasmic reticulum. So this is called KDEL. Another example is called, the second is called mannose 6-phosphate. This is also a mark, and this mark implies that this protein must go to the lysosome. All the lysosomal enzymes modified in this way, because I have mentioned in my first class of this vesicular trafficking that in the oligosaccharide or glycosylation there are mannose sugars are present. Mannose have 6 number of carbons or 6 carbon. At that 6 carbon, phosphate group are added by a special mechanism in acetyl glucosamine transferase. I am not going into details, but mannose 6-phosphate tag, these are all tag. If any proteins have this tag mannose 6-phosphate, that means it will destined to reach to the lysosome, and this receptor also present in the trans-Golgi network. You must remember, in case of KDEL receptor, this is present in the CGN. For the mannose 6-phosphate receptor, that can catch that will actually attach with this protein, is present in the TGN. And another example is plasma membrane. Some receptors are also present in the plasma membrane. Why? Because during some lysis process, lysosomal enzyme can actually secrete it out, and this enzyme is actually retrieved by this receptor present in the plasma membrane. Okay, so these are the helper protein. GTP binding proteins helps. If you I'm actually referring you two books. One is Lodis and Bruce Albert, latest edition. So you can see how this SAR1 helps in the coating of the vesicles. So I am giving you a very simple way. So let's suppose this is the endoplasmic reticulum. When budding occurs, this is a endoplasmic reticulum. When budding occurs, bud actually occurs like this. A bulging like structure actually form. Bulge like structure. This is actually pre-forming vesicles. Now, to pinching off, to cut these vesicles from the ear, this dynamin protein is important. Dynamin protein actually cover this neck region, and actually by the help of GTP hydrolysis actually pinch off these vesicles and actually cut the vesicles from the endoplasmic reticulum. So these are the plasma membrane, nothing but plasma membrane. Now the vesicle is complete. Now this vesicle is only made up of plasma membrane, very delicate and fragile. So they should be coat. Now SAR1 protein, this, if it is a SAR1 protein, it has a tail-like structure. When it is associated with GTP, the tail is actually protrude, open. But when it is not attached with GTP, let's suppose this is not GTP, then the tail is actually backward. If it is GDP, let's suppose this is GDP, and it is GTP. When it is attached with GTP, it will bind with these vesicles and actually helps these coat proteins to coat the protein. And also this SAR1 has an intrinsic property for the GTP hydrolysis. It can degrade this GTP into GDP, and then it actually helps in the removal of the coating, because unless or until the coats are removed, they cannot fuse. For the fusions, the coat should be removed. So one is vesicle budding and there is vesicle fusions. So in this class I have discussed about only the vesicle budding. In my next class I will talk about vesicle fusion. Continue watching.